Thank you, Dora. And there we go. Show my screen. And hopefully you're going to tell me if you can't see my screen or anything wrong, send me an email or a chat thing, and so I know what the heck's going on. So my name's Bob Borden. I'm going to be speaking about anaerobic bioremediation at DNAPLs. And just to get started, I have to do my sort of official disclosure thing here. I'm a principal at Solutions IES. I'm also a professor at North Carolina State University. Principally do soil and groundwater remediation research, but NCSU is not sponsoring or endorsing this presentation, and you get no free football tickets. So with that said, I'm going to minimize these things and move on. I think I'm going to move on. There you go. Okay. Webinar outline. First of all, talk about bioremediation objectives. A lot of this webinar actually is not my own research. It's really, to a certain extent, a lit review of a lot of other people's work, and then I'm going to show you at the end some of the stuff we've done to try to overcome some of these processes. And in my opinion, there's really three big factors that have an influence on anaerobic bioremediation DNAPL. One is toxicity of the contaminant itself to the bacteria, competition for electron donor, pH issues, and slow mass transfer. And as I said at the end, I'll show some of our own work, or at least, actually not my own work, Kitty Hyartal's own work, but what the heck, I'll take credit for it too. She was my grad student. And then some lessons learned. Okay, so before we get started, just want to sort of identify some ground rules. I think you're all aware anaerobic bioremediation has been used at thousands of sites for plume control of chlorinated solvents, for chlorate, explosives, lots of things. Very well established process. There's good design rules. That's not covered in this presentation. So I'm not looking at the down gradient plume, low concentrations. We're really strictly focused on the source area here. I'll speak a little bit about what I'm going to call low to moderate concentration source areas. In general, treating them is not that different from plume treatment. It's, at least for the high K zones, it's relatively easy to treat. The low K zones, it's just simply difficult to distribute donor, and so the effectiveness of treating the lower K zones is challenging. But you can put, well, for me, emulsified oil in the high K zones as the contaminants diffuse out of the low K zones into the high, it gets treated. So again, low to moderate concentrations, at least I personally feel we're okay on. We're pretty good at treating source areas with that. The real focus of this presentation is on DNAPL. I mean, pure phase, little droplets of TCE or PCE. As what I'm going to, at least I think, show you in this presentation is remediating DNAPL is quite challenging. I think we made some progress, but there definitely are some technical challenges. Okay, so before we get started, just baseline. The baseline I compare it to is pump and treat. You can certainly go out, you can put a pump and treat system in, you extract groundwater, the groundwater flows past the DNAPL, it dissolves a portion of it, and then you pump it out. Significant O&M costs. The big problem is chlorinated solvents are not very soluble, and so as the groundwater flows past the DNAPL, only a small fraction of it dissolves, which means you have to keep pumping and pumping and pumping for many, many years. I think this is sort of the baseline that people talk about. And if we're talking about the remediating a source area, what you really want to do is compare anaerobic bioremediation to this baseline and how much better it does than this. What is anaerobic bioremediation? At least for a lot of people, it looks pretty similar. We got an injection well, we inject some food, it flows past the DNAPL, we dissolve away a portion of the DNAPL, it biodegrades, we pump it out, and we pump it around again in a circle. Conceptually, it doesn't look all that different from a pump and treat system. I think the difference is anaerobic bioremediation systems, at least in my opinion, the recirculation systems generally have significantly higher O&M than pump and treat. First of all, you have to pay for the substrate. There's more monitoring, process control, and then there's just more logistical problems with plugging of piping and biofouling and stuff like that. What do you get from that? Well, really you get two possible benefits. One is you can transform the contaminants into non-toxic end products. While that is certainly possible, that's probably not the primary end result when you're actually bioremediating DNAPL. The other thing is you can actually clean up the sorts faster than you would with pump and treat. And I think that's the primary objective most people have focused on. Okay, how does that work? This is some lab studies from Perry McCarty and one of his students, Yang. And what they did is they took a laboratory column and they put little droplets of pure PCE in it, and then they pumped, I think in this case it was pentanol, through the laboratory column, and they bio-augmented it, and they monitored. And what you see here is you start off, concentrations are low. Over time, let's see, the dark circles are PCE, the dark squares are DCE. So we're within about 4 months here, basically, we're producing a lot of DCE. And if you sum up the DCE plus the PCE plus the TC plus the monochloride all up, you get this solid line up here. Well, PCE has got an aqueous solubility of about 1 millimolar. Total chlorinated ethanes here is about, I don't know, 4 to 5 millimolar. So essentially you've increased, in a weird way, the solubility of this mixture by a factor of 4 to 5, or at least its dissolution rate by a factor of 4 to 5. So presumably you would clean up a DNAPL source area 4 to 5 times as fast. So that's the good news. The bad news is you can see here we really haven't, we've converted PCE to cis DCE. There's still a lot of DC coming out, so it's not a non-toxic end product. It may be less toxic, but it's certainly not non-toxic. What you would need to do if you were actually in the field doing this is there would need to be some other treatment technology further down gradient. Could be a groundwater extraction well, or for us, we'd probably go down gradient and we'd put an EOS barrier so that DC would flow through the barrier and it would get treated. Okay, here's a bunch of studies. There have been a number of studies people have done on chlorinated or bioremediation of chlorinated solvents. Yang and McCarty, that first one, they had a, well, if you remember, the maximum improvement here was about a factor of 5, but then early on it was maybe 2 to 3. So if you looked at a cumulative, this might have over the entire project or experimental period, it might be a factor of 2 to 3 increase with a factor of 5. If you look, here's a series of column studies, dissolution enhancements, well, the maximum was 5. Carr et al., he had a maximum of 14, a cumulative of 14. A lot of here, okay, Yang and McCarty using pentanol, oleate, and olive oil, cumulative improvements of 1.6, 2.3, 1.9, sleep 1.7, but a maximum of 3. So you can see here these kind of numbers. Some cases you could get really high maximum improvements. Ben Amos got up to 13 or for the maximum, but typically we're seeing cumulative remediation improvements of anywhere from 2 to 3, maybe 4. So not, I mean, good improvements, but not giant improvements. The other thing you notice here is there's a couple descriptions here. Carr et al. They actually weren't bioremediating pure PCE. What they did is they mixed 1 part PCE with 3 parts tridecane. The reason they did that is PCE is toxic, and it killed them. And so they diluted the material with basically a inert hydrocarbon that reduced the effective solubility by, I guess, they're about a factor of 9. Well, so what you have to remember is, okay, there was a cumulative improvement of a factor of 14 over something that was already diluted out by a factor of 9. So really, the real improvement might be 1.5. Ben Amos, he did an initial column study. He tried neat PCE. I'll show you those results in a little bit. But basically, the PCE killed all the bacteria, so he didn't get any improvement. He went back. He diluted that material, 1 part PCE with 3 parts hexadecane, and it worked very, very well. And he got a factor of 4.6 improvement over the baseline, but the baseline was 25%, was only 25% of the PCE solubility. So he basically got back up to what he got with just pure PCE pumping. You always have to remember that dilution effect. But this toxicity is real, and people worry about it significantly. Okay, so how does this really work? You know, why are we getting the solubility enhancement? What happens is, as groundwater flows past a pure PCE DNAPL, the PCE will dissolve. Once it gets up to about 170 milligrams per liter, or 1 millimole, the groundwater is saturated with PCE, and so no more can dissolve. If that PCE dissolves or biodegrades, then more can dissolve. Now, if you produce enough TCE or cis-DCE, they'll dissolve back into the NAPL and sort of reduce your treatment efficiency. But the key thing is, cis-DCE is 1/36, or 36 times as soluble as PCE. So really the back partitioning of cis-DCE is probably less important. That's basically why this process works. We're converting a less soluble material into a more soluble material that can then be more easily pumped out. If you converted all the PCE to 100% DCE, in theory, you'd get a factor of 36 improvement in flushing efficiency. You saw from those previous results that it was more common. We got a factor of 2 to 3. So not nearly what the maximum possible is. My question is, why? Okay, well, I'm going to try to answer that. This is actually an experiment was done by Cedergren back in 2002. It was not DNAPL. This is actually an LNAPL. He had I don't want to call it a column. It wasn't exactly a column, but he's got a column here with a series of sampling ports along the top. He pumps water on one side, water out the other. It flows through the sand. And he actually sort of built this little reservoir on top of the column. And he filled that reservoir with LNAPL. Well, since it's lighter than water, it stayed up there. And so what would happen is water would flow by there. The LNAPL would dissolve, and it would biodegrade, and it would keep flowing on its way. But now what you have to remember is, okay, so as the water is flowing past this LNAPL here, it's dissolving. If the contaminant biodegrades before it passes the LNAPL, more will dissolve. But suppose toluene here dissolved out of the LNAPL, it flowed down, It flowed down here, and it biodegraded somewhere down here at the end of the column. Well, that's actually not going to enhance dissolution because it's no longer in contact with an apple. To enhance dissolution, the biodegradation has to occur right here. And it has to occur fast. The faster it biodegrades, the faster more can dissolve. And Cederin came up with a relationship. I'm not going to go through all the derivations, but it was in a paper in 1994. And basically what he showed is you could characterize this sort of the enhancement factor, how much improvement in removal based on a Domkohler number. And the Domkohler number had to be greater than 1. Well, okay, what's Domkohler number? It's the degradation rate K times the contact length, the length of that DNAPL zone, divided by the velocity. Well, length divided by velocity is contact time. So you need have a degradation rate times contact time greater than 0.1. As you look here, even a contact time of 1, well, yeah, maybe if that's an enhancement factor of 1, that's really no enhancement. This is 2. So a Domkohler number of 1 gets you a 1.5 improvement. But if you can go up to a Domkohler number of 10, now you got a 3-fold improvement. 100, you've got a 10-fold improvement. And 1,000, you know, maybe even a 30-fold improvement. So the key thing to get this process to really work well is you need, well, either long contact times or high degradation rates, or better yet, both. If you have a short contact time and a low degradation rate, it's really not going to do any enhancement. Let me go back. So if you had a low Domkohler number, the groundwater here would get saturated with toluene. It might flow down gradient, and the toluene might get degraded, but it wouldn't actually accelerate removal of this NAPL. Hope that makes sense, and if it doesn't, come ask me a question later. Okay, so the key thing is we not only have to degrade PCTC, we have to degrade it fast. So now there's some problems with that. Number one, PCTC is toxic. It will inhibit the bacteria. 2, there are just technical complications of getting the electron donor to the DNAPL. 3, as we're doing this dechlorination, we're going to produce hydrochloric acid. And most of the point, the hydrochloric acid is produced where the bacteria are. So you also have essentially a mass transfer limitation of getting the acidity away from the bacteria and not inhibit them. And then finally, just surface area mass transfer limits. We said DNAPLs dissolve slowly. Well, if the DNAPL dissolves slowly, you can't biodegrade it any faster than it dissolves. So there's an inherent sort of physical limitation on that. Okay, let's talk about toxicity. There's been a bunch of studies here. I'm just reporting a couple of them. Ben Amos, he reported basically complete inhibition when PCE was greater than about a half a millimolar for 4 different pure cultures. Melanie Duhamel, working with KB1 culture. Now the key thing is this is not inhibitory concentrations. At these levels, she was able to get complete dechlorination to ethene. So 0.8 millimolar of dissolved PCE in a batch microcosm was completely degraded, but that was when she added a lot of biomass. So once the PCE degraded a little bit, it gets less toxic. When it degrades more, it gets less toxic, and the process went on. Same way, 1.5 millimolar for TCE and 1.4 for vinyl chloride. That's total inhibition. So basically at these levels, they were not able to get degradation. There's another way to think about toxicity, though, and this is an idea that Sabot, I hope I don't mess up his name too badly, Bolowski and Luciprini did a study, and they looked at toxicity, and their argument was one way to think about toxicity is just sort of death rate. If you're in a toxic environment, it's harder for the bacteria to live. It takes more energy to survive. So essentially, they're going to die quicker. And what they came up with is that at a PCE concentration of 0.2 millimolar, the effective decay rate of the bacteria is increased about tenfold. Well, if your growth rate was only 10 times the death rate to start with, which is not uncommon, at this point here, it's not like they're totally inhibited, they're just dying faster than they grow. So over time, the bacterial population will decay away, or maybe just simply they won't grow up as fast. And it'll take longer for them to grow up, they'll be inhibited, and then just the process takes longer to go. And you can see these numbers are quite a bit lower than some of the complete inhibition numbers. So at least based on their work, even a PCE concentration of 20% of solubility is going to slow the process. It's going to make it more difficult to get high bacterial numbers and really excellent decay rates. And that's illustrated here. This is that, I just wanted to show you a picture. I mentioned it before. These are the initial results from Ben Amos's column. He added pure PCE to the column. He pumped water with a bio-augmented with a culture that could degrade PCE. And you can see here, and within 1 pore volume, the effluent concentration jumped up to 1.2. Some people say the solubility is 1.2. Well, here it looks like it's 1.2. And it basically stayed there. So there's it's just the water saturated with PCE. Now you can see here, he did make a little bit of cis-DCE and a little bit of TCE, but that's over on this axis. He's discharging 1,200 micromolar PCE and 7 micromolar of DCE. So he's increased the effective solubility from 1,200 to 1,210. A pretty trivial enhancement. But this was, again, pure, neat PCE. When he went back and he mixed the PCE with hexadecane, it worked great. And he's got a wonderful paper. He got enhancement. But basically, he had to dilute away some of the PCE to reduce that toxic effect. Here's a study by Mark Harkness. What they did is he had, again, a column. He saturated it with DNAPL, or at least the inlet of the column with DNAPL, let it drain the DNAPL out so it was at residual saturation, and they injected an emulsified vegetable oil from SRS. Now, one thing I have to add is, in this study, they tried to use as little EVO as possible, because I'm not sure. I think they were basically just trying to see what kind of efficiency they could get. And what you see here is, well, basically it didn't work. Sorry about that. Basically, we're coming out. The column is discharging, 6,000 to 8,000 micromolar of TCE. You can see down here a little tiny bit of DC coming out. What they attributed that to is actually, as the experiment went on, you dissolve away some of the TCE from the inlet of the column. So actually, they started getting some degradation at the very, very inlet of the column before, where there really wasn't much TCE anymore because it had dissolved away. But in the really, neat DNAPL zone, they really got nothing. Key thing I'm going to show you some later results that we did that look very different from this. We added a lot more EVO than they did. They were trying to minimize it. We were not. And And I personally feel like that makes a big difference. Okay, so calamity toxicity is a problem. Another issue is delivery. This is a sandbox study done by Brent Sleep and some of his colleagues at the University of Toronto. They did two different sandboxes, actually, one with no bioaugmentation, one with bioaugmentation. Here's what the box looks like. The black dots are sampling ports. This little rectangle here is where they injected some neat PCE, and now what they're going to do is they're going to pump an alcohol solution. I think it was a mixture of methanol and ethanol into the inlet. It's going to flow past, and they're going to get degradation in the DNAPL here, and then it's kind of smeared around here. These are results for the bioaugmented box. You can see before they bioaugmented, water ran along, and then eventually they got up to about 0.3 millimolar of PCE. Here, they bioaugmented almost instantly. The PCE dropped down to very, very low levels, and they made a bunch of DCE. And then over time, gradually, the DCE started to decline, and they started getting some degradation products. So they got some enhancement, but, not great. They got an enhancement, I shouldn't say that. If you look at total ethines removed, that's just the sum of PCE, TCE, DCE, vinyl chloride, and ethene. What you see here is the two boxes. I think the dark symbols are box 1, the not bioaugmented. Box 2 is the open circles. You can see here, with or without bioaugmentation, they're tracking pretty similarly. As soon as they bioaugment, they get this big jump. That's the bioenhancement factor. They basically doubled when they bioaugmented their removal efficiency during that time period. And you can see it stays high for a while here, and then eventually declines. This actually looks, if you just look at this data, it looks really good. And eventually it declines down to, really low numbers where basically there's no ethynes coming out anymore. And it looks like they cleaned up both boxes. Well, the problem is that's not actually what happened. When they went back and did the mass balance, at the end of almost 900 days of operation, they had removed 65% of the PCE in the bioaugmented box and 39% in the box without bioaugmentation. But, well, if they removed 65%, that means 35% is still there. Yet nothing is leaching out. What happened? Well, what they think probably happened is that, remember, you're pumping ethanol and methanol in the inlet. The PC's over here. What they could see is they started to produce a lot of methane in this region, up gradient of the DNAPL. And essentially what was happening is, basically they were wasting their substrate. It was all fermenting the methane before you even got to the contaminant. And worse yet, they were making, not really sure, but they seen it was fairly clear that substrate was not getting to the contaminant. And in fact, there was probably some permeability changes. It might have been due to biomass. It might have been due to gas bubbles. But the water was sort of flowing around the contaminant, not through it. It doesn't flow around. It doesn't dissolve. You don't get degradation. I believe one of the things they did just to verify this, they went back and they actually injected the substrate directly into this zone here where they put the DNAPL, and they got a spike and an improved efficiency for at least a short period of time. In the field, if you knew exactly where you spilled your DNAPL and you could put a well there and inject into it, that might work. Unfortunately, we don't usually know where it is, so that it proves their point, but I'm not sure if in practice you could do that. And you can see that here with, big, large amounts of methane being produced and I believe this is just aqueous phase methane, so there's probably some bubbles produced too. Okay, so delivery of substrate to the contaminant can at least also be an issue. Okay, another issue that we have to concern ourselves with is acidity production. As you go from PC to TC to DC and so on, what you're going to do is you're going to convert 1 mole of PCE to 1 mole of ethene and 4 moles of hydrochloric acid. If you want to just do it on a mass basis, if you burn up a kilogram of PCE, you made about 0.9 kilograms of hydrochloric acid. That does not include any acidity associated with carbon dioxide, with the fatty acids you're producing. So you can produce a bunch of acid. And the thing I focus on is you're producing the acid right at the surface of the bacteria. So even if if you measure the groundwater, the pH is 6, it certainly could be 5.5 or 5.0 at the surface, right where the bacteria are, because you're undoubtedly not measuring that in your monitor wells. I'm sure you've all seen these kind of graphs before. This is from Ashley Eady, and she showed for degradation of TCE, got pretty good TCE degradation. The best was 7, but even at 6 or even at 5.5, degradation was slower, but you still got it. So for the best degradation, we probably want a pH of 7, but 6.5 is probably fine for pretty good degradation. Conversely, for vinyl chloride to ethene, these bugs are much more pH sensitive. That may be less of an issue for us because we're probably not going to make much vinyl chloride anyway, because essentially what typically happens to these sites is mostly you convert PC and TC to DCE, and the DCE doesn't degrade until some distance further down gradient. Okay. I developed a little mathematical model here. I'm not saying this is, it's certainly not the most sophisticated one, but I did this to sort of illustrate what I think are some of the issues here. And the way I represented it, I said, first there's an initial step where TC dissolves. So the change in aqueous TC concentration equals a mass transfer coefficient times the surface area of the DNAPL droplets times the difference between the TC solubility and the TC concentration in water. That's the driving force, and that adds TC to groundwater. And then TC degrades by this term here. Loss of TC loss of TC here increases the amount of DCE, and then as the DC degrades, you make vinyl chloride. Vinyl chloride degrades, you make ethene. Okay. And these are sort of my base parameters here. I'm assuming a TC degradation rate of 10 per year and a DC degradation rate of 5 per year. With no degradation and a 50-foot wide DNAPL zone, what we'd expect to happen is the TC concentration to rise up quickly as it's flowing past the DNAPL. As it gets closer and closer to saturation, the rate slows. But by the end of the 50-foot wide zone, basically we have groundwater saturated with TCE. Now, if it keeps flowing down gradient and there's no degradation, this won't go down. It just stays high. If we go up and we bioaugment and we bioremediate and we add a substrate, what's going to happen is TC will start to dissolve and increases and increases and increases. And then what happens is, as you get out here, now the TC is degrading, producing DCE and vinyl chloride and ethene. So while it's still dissolving, it's also degrading. So whereas here at 50 feet, we're up at, 8 millimolar. Here at 50 feet, well, really at 25 feet, something like that, we plateau out at 5.5 or 6. So we're undersaturated with PCE, or TCE. It keeps dissolving. And so the net result is, when you sum up all these terms here, by the time we get to the end of the DNAPL zone here, we've got 21, something like that. millimoles of total ethynes in solution. Here was 8. So we just got a factor of 3 enhancement in dissolution, roughly, maybe a little less than a factor of 3. But that worked because it was degrading while it was still in contact with the NAPL. Okay, so I did some simulation trying to see what the different parameter effects have. Here I've got 2 graphs. I've got a high mass transfer rate, and I don't really know if that's high, but relative to degradation. So a mass transfer rate of 10 per year, and a TC degradation rate of 1 per year. The TC dissolves in the water, it gets close to saturation, but since degradation is slow, I've only produced a little bit of DCE here, so that I'm only getting up to a maximum of 10 millimolar. Conversely, if I use a degradation rate, a TC degradation rate 20 times as high, now as the TC dissolves, I'm getting really rapid production of DCE, rapid production of vinyl chloride and ethene, and so now I've increased the effective solubility, or the amount removed, up to 25 something. This just illustrates you need high degradation rates to do much good. If you have slow degradation rates, even though the TC is going down here and I'm making stuff, I'm downgrading to the source, so I can't really accelerate removal of the source since I'm already past it. Now, I do the same simulation with a slow mass transfer rate. Here, with a slow mass transfer rate of 1 per year, I never actually even get up to fully saturated. There's really little benefit. I'm not even saturated here. If I have fast degradation and with a slow rate of mass transfer, what's good is very little discharges down gradient. By the time I'm 150 feet down gradient, all the contaminants are converted away. But I've only dissolved a little bit more than 5 millimolar. There's really almost no improvement in removal. At this point, bioremediation isn't really doing any good. I mean, it's limiting downgradient migration, but it's not increasing the cleanup, because this stuff just doesn't dissolve fast enough. It has to dissolve before it can biodegrade. If it doesn't dissolve, you can't do any good. Okay. So what we've done is, I feel like what we've done is we took those lessons learned that the rate of degradation is limited by, well, one, toxicity, contact with the substrate, pH, and try to work and develop a process that would at least minimize those effects. So what we do is we flush emulsified oil and pH buffer actually through the DNAPL zone. What that's going to do is the TCE will or the PCE here will partition into the oil, and it'll make a mixed PCE soybean oil droplet. Those droplets have a lot of surface area for bacterial growth. We reduce or eliminate the mass transfer problem, or the wastage problem, before the substrate was being consumed, before it reached the DNAPL. Well, if it's already mixed with the DNAPL, there's no waiting. So that droplet's going to simultaneously dissolve away. It's going to release PCE and soybean oil. We feel it's really important that we distribute the buffer through the treatment zone also. So while the bacteria are producing acid, there's buffer very, very close to them. So it neutralizes the pH as they're producing acid. The other thing is, if we could mix 90% oil with 10% PCE, we'd make a new mixed NAPL, which would be 10% PCE, and should release dissolved PCE at 10% the solubility. If we reduce the effective solubility by a factor of 10, we're going to reduce the toxicity by a factor of 10. Or maybe not a factor of 10, but we're going to reduce the toxicity. This work was done by Kirsten Hyortal, and they're smiling at me because I always destroy her name. I'm sorry, Kitty. And I'll show you some of the results. So what she did is she took a lab column. You can see here the red nasty stuff here. That's PCE dyed with red dye. We injected a ml of PCE in it, and it sort of flowed down and saturated this end of the column. And then we pump a mixture of emulsified vegetable oil and magnesium hydroxide up through it. If you look at the picture, you can see that kind of white stuff. That's the EVO and magnesium hydroxide. And then we immediately bioaugmented. We did 3 treatments, one with EOS Pro, one with this new product or new material that Kitty developed. It was EVO plus 5% magnesium hydroxide, and there's a control. One thing I had to note, Mark Harkness and Angela Fisher did an experiment similar to this. They didn't use the pH buffer. We're adding a lot more EVOs than they did. They tried to add the smallest amount possible. We wanted to add a large excess. So here are some initial results. Okay, so this is during the initial flush of EVO through the column. And what you can see here, we're starting out with, really much lower PCE concentrations. Aqueous solubility PCE is about 1,200 micromolar. We start flushing the EVO, we get this big spike. We're up to like 15,000 micromolar, so factor of 8 above the aqueous solubility. So what that is we've actually got droplets of soybean oil flowing out the end of the column. That's, well, it's 90% soybean oil and it's 10% PCE, or I'm not sure the ratio. But there's actually PCE in those little oil droplets that are coming out. So we get a big spike. We did that. I wanted to prove that you could actually get the PCE to dissolve in the oil and it would flow 5 feet down gradient. In the field, you wouldn't add this large excess so it wouldn't migrate nearly as far. But I had to get it to come out of the column to basically so I could give this presentation and prove that we could increase the solubility. For MH5, the MH5 doesn't transport as well, so you didn't see as big a spike, but you clearly did see some spike. So that was the initial injection. I think this took like 4 hours, and then we bioaugmented. So we really flushed the EVO through that NAPL zone very, very quickly. And here's some of the results. You can see with EOS Pro and bioaugmentation, initially we get a big spike in DCE, and then it starts to decline off. There's a little bit of PCE coming out. Initially it actually dropped. It reached a minimum right about here, about 120, 30 days. And then it actually started to go up. This column test honestly was a failure. Yeah, we got some DC production, but what happened here, as you can see, is we started producing DCE. The pH, the open symbols here is the EOS column. The pH started to drop right here where the PC is at its lowest level. We've got a pH in the column effluent of like 5.5. A pH of 5.5 plus 0.2 millimolar PCE killed the bacteria. Or it didn't kill them, we're still getting some degradation, but they're just clearly not happy. And they're not doing what they really need to. By contrast, when we add EVO plus magnesium hydroxide, you get a little bump initially, but here shortly after injection, we have 99.9% conversion. The only thing that comes out of this column is DCE. And you can see here by the end of the experiment, we're releasing 1.2, 1.5 millimolar of DCE, even though we've mixed the DCE or the PCE with vegetable oil. that should have essentially reduced the aqueous solubility. We don't have a giant enhancement, but I haven't shown you here in the control columns where only basically the control columns went up and they released a maximum about 0.3 millimolar of PCE. So we've got a factor of 3 or 4 increase in effective solubility over that alone. One thing to note, well, we just weren't very smart and didn't do our calculations right. We didn't add enough magnesium hydroxide in this column. By this point here, about 300 days, we were doing a mass balance on magnesium, and like 99% of the magnesium we added was flushed out of the column here by about 300 days, or maybe 250. And you can see here in the MH5 column, we maintained a nice neutral pH until here about 250 days, and then we get a pretty abrupt drop. Basically, we had depleted all the magnesium hydroxide by this point, and the pH dropped down. So now we're down here at a pH of about 6. 6 is not good, but it's not nearly as bad as 5.5. Plus the fact at this point there's virtually no PCE in this column anymore. So we've dramatically reduced the PCE toxicity by the time the pH buffer starts to get depleted, and so clearly the DC production is continuing to increase here. Okay, so lessons learned. Yes, it is definitely possible to bioremediate pure DNAPL. Typically, what you're going to see is at least in the DNAPL zone, you're going to produce DCE if you're doing it right. Down gradient, that DCE can further degrade to vinyl chloride and ethene. But as long as there is PCET or TCE available for the bacteria, they will preferentially use that over DCE, and you really have to kind of get rid of all that first before you get good conversion. 2, to rapidly deplete the DNAPL, you need high mass transfer rates and rapid bacterial growth. It can be done, but it's challenging. There's been certainly you can stimulate, you can bioremediate DNAPLs, but there are complexities associated with toxicity, competition for electron donor, low pH, surface area, and it's bioremediation of neat DNAPL is technically more challenging than just a dissolved plume. And that's all I have. And I'll turn it back over to you, Dora.
All right, thank you, Dr. Borden. That was a great presentation. We do have a couple of questions. The first is, with the pH drop that was seen in the MH5 column, you were still seeing an increased DCE degradation rate. What do you think caused that?
Go back. Honestly, we don't really know. The explanation I have is, number one, if you look down here, it dropped pretty low, but actually it bumped up a little bit here. We're actually at this point about 6.1, 6.2. That pH is not that bad, especially for PCE conversion to DCE. The other thing is, based on what I haven't shown you is the chloride data. By the time we're here, it looks like we've dechlorinated a lot of the PCE. And it's possibly a fair amount of this DC that's coming out here was actually produced sometime in the past and is actually sort of sorbed inside the vegetable oil. So we believe that the PCE toxicity has been significantly reduced by here, and that's why we're getting this gradual increase, or at least that's the best explanation I have. Any other questions?
Yes, there's another question. What exactly were you bio-augmenting with?
What culture?
Yes.
I think I said somewhere. What do we use? Yeah, Bac9 culture provided by EOS Remediation. I had to put that plug in there.
Okay, let's see. From slide 24, the model with a high biodegradation rate indicates a concentration of ethene up to 25 millimolar. Since the saturation concentration of ethene in water is 4 millimolar, how do you explain the divergence from the predicted results to actual concentration?
I have to. I'm not advocating that this model is a appropriate, detailed representation of what would actually happen. If you actually produce that much ethene, you get gas bubbles coming out. Yeah, this is way more than, and we've not included basically degassing of ethene in this. I've also not included the fact that at these high DCE concentrations, you would get some DCE partitioning back into the NAPL, and that's not included here. This is a very, very simplified representation. What I just wanted to point out is the sort of, I couldn't find any good illustrations in the literature of sort of the effective degradation rate, and I was trying to just point out that, you know, you really need to get good degradation rates to have this work. Slow degradation doesn't do much for you.
Okay. So what's the best way to calculate the volume of buffer that is needed to be applied?
We do it process of things. Calculate the amount of acidity that's going to flow in from up gradient. Calculate the acidity associated with the soil. Calculate the acidity associated with hydrochloric acid you're going to produce during the bioremediation process. And also calculate the acidity associated with CO2 you're going to produce during the bioremediation process. Add them all together, and that's the amount of buffer you have to add. One thing I have to add that's a problem with this that we have not solved yet is if you're producing a lot of, ideally what you'd like to do is you just go out there one time, add all the magnesium hydroxide you need for the life of the project. Problem is you're adding a bunch of magnesium hydroxide before the hydrochloric acid is produced. If you add 100% of that magnesium hydroxide at the beginning, there is a possibility you add too much, and now you make the pH too high. Because, if you add, 1,000 pounds of magnesium hydroxide, and you don't produce the acid till a year later, well, you could overshoot. And that's, we have not totally solved the pH issue. We feel like the magnesium hydroxide works better than other alternatives, but particularly with a DNAPL like this, you may need to add more buffer, every 6 months or a year, go back and add more buffer, because adding all that at the beginning may be a problem.
Okay. The next question is, you presented many examples of laboratory column studies. Are you aware of any field studies showing success of DNAPL bioremediation?
The guys with the SABRE group in the United Kingdom, it was a consortium of industries. EPA was involved. If you search SABRE, I guess, on the website, they did a very detailed study, and they got some really good results. Unfortunately, that the paper by Mark Harkless was part of that effort. They've got their field results on the website, but it's really kind of a short summary, and I am waiting tensely to get the full paper when it comes out, but it's, journal publishers take a long time. That's probably the best documented case. I certainly know of other people that have bioremediated DNAPL. usually not in the level of control required for a nice presentation. It's, we squirted stuff in, we made a bunch of cis DCE, but actually comparing removal rates to controls, you have to have a control to compare removal rates. Next, any other questions? Well, we've got a pilot study going on right now that's a field bio-DNAPL project, but we haven't published those results yet.
Okay, and the next question on that, the follow-up, is what are effective approaches to help control biofouling that can occur with bioremediation systems?
I think operating experience is that if you do emulsified oil, biofouling is much, much less of an issue because basically it takes weeks or months for the bacteria to grow up around the injection well. So if you go out, you inject the EVO, it flows out away from the injection well. As long as you chase it with a lot of water to sort of clean up your well, the well won't biofoul. And then if you come back 3 or 4 years later, you inject it again. And we don't really get that problem. With soluble substrates where they're continually recirculating, it's much more a problem. There's actually a good report written for ESTCP. It's on their website somewhere, and it talks about methods for controlling biofouling. They do a number of things. They, pulse the injectate. So they'll inject a concentrated pulse, then chase it with water, concentrated pulse. Sometimes they use chemical inhibitors, citric acid, low pH to inhibit the growth around the well. Sometimes they just have to go clean the well every, once a week or every 2 weeks. There are things that work, but a lot of them are relatively labor intensive.
All right, 2 last questions. The dilution of a large DNAPL pool can take years under field conditions. So do you think the cost of the dissolved NAPL approach that you were describing could be significantly higher than if you did a direct DNAPL removal?
Direct DNAPL removal, like dig it up and haul it away. If you can get to it, it's not too, there's a lot of other very good competitive technologies that may be more appropriate for really, truly pooled DNAPL. Thermal, excavation. I'm not actually a real big fan of doing bio after ISCO just because of the changes, but some people do that. So there are, for pooled DNAPL, yes, there are many probably superior alternatives. The way we think of it's best suited is that if you can find the hot spots, remove them either by thermal or excavation or something else. But there's a lot of cases where we believe there are droplets of DNAPL scattered around this large area. We just can't find them. And doing thermal over a large area sometimes is cost prohibitive. So this is one alternative where you could treat, I'm going to say, lower concentrations of DNAPL, but not, pools. Might be the sweet spot for bioremediation.
Okay, and the last question, do you know of any researchers that are evaluating methods to help enhance the biological resistance to PCE toxicity?
Don't know. I'm not aware of anything. Part of the problem is just, I know people working on developing enriched cultures that can handle lower pH, but part of the issue is just the membrane around bacteria is made of fat or fatty acids. PCE will dissolve the cell membrane, and that would take some fairly sophisticated engineering to solve that problem. But well, I'm not that kind of microbiologist, so I won't say what they can do.
All right. Well, thank you very much, Dr. Borden. It was a very informative presentation, and thank you all for attending today. If you have additional questions or you would like further information, you can contact us at info@microbe.com, or you can directly contact Dr. Borden. Again, his email address is rcborden@solutions-ies.com. Thank you, Dr. Borden, and thank you everyone for your time. Have a great day.
Thanks a lot, Dora.
Thank you.